Good morning, students. Today we will talk about extracellular matrix part 2. In last lecture, we learned about functions and components of extracellular matrix, in detail about the collagen protein. Now we will learn all the remaining extracellular matrix proteins. Now next is structural protein is elastin. So, elastin is a major protein component of tissues that require elasticity, such as arteries, lungs, bladder, skin, and elastic ligaments and cartilage. So, it is a polypeptide chains which are cross-linked together to form a rubber-like elastic fiber. Each elastic molecule uncoils when fiber is stretched and recoils spontaneously as soon as the force is relaxed. It is composed of soluble tropoelastin protein. Mainly it contains glycine and valine and modified alanine and proline residues. So tropoelastin is a 750 amino acid long protein that is highly cross-linked to form an insoluble complex. Elastin mainly it is secreted by connective tissue cells as soluble tropoelastin into extracellular matrix. It forms cross-linkages with each other which are catalyzed by lysyl oxidase. It forms an extensive network of elastin fibers and sheets. In the figure, you can see when it relaxes, elastin molecule relaxes, it recoils, and when it is stressed, it uncoils. And you can see single elastin molecule is there, this one, and after cross-linking they are forming cross-linked molecule. As we studied collagen, we have seen elastin. We can see the difference. What is the difference between these two structural proteins, that is collagen and elastin? So collagen have many different genetic types while elastin have one genetic type. Collagen, it is triple helix. Elastin, no triple helix. It randomly coils conformation permitting stretching. In collagen, they have glycine XY repeating structure, but as such there is no repeating structure in elastin. Presence of hydroxylysine carbohydrate containing in collagen, no hydroxylysine or no carbohydrate in elastin. There are aldol crosslinks in collagen, desmosine crosslinks are there in elastin. Presence of extension peptides during biosynthesis we had seen in collagen. Thus, there are no extension peptides present during biosynthesis. The next is the fibrillin. Fibrillin, which is a glycoprotein secreted by extracellular fibroblast. It is found in jonular fibers of the lens, arterial wall and periosteum. So there is a genetic disorder, that is Marfan syndrome, which is associated with the fibrillin molecule and protein. It results from mutation in fibrillin gene and it is an autosomal dominant disease. It is present in Marfan syndrome. There is skeletal abnormalities, ocular changes and cardiovascular lesions are also there, as you can see in this picture. Skeletal abnormalities you can see in the limbs, ocular abnormalities and the cardiovascular abnormalities are also there. Next is fibronectin. It is a high molecular weight glycoprotein. It is attached to cell membrane by membrane spanning receptor, that is integrin. Now look at the functions, what it do. First, it helps in wound healing. How it guides immune cells to wounded areas and thus promote wound healing. Second is clotting process. It helps in clotting process. It links to fibrin and helps in the clotting. Next, cell adhesion, growth, migration and differentiation. And next function is, it is found in normal human saliva, so it prevents colonization of oral cavity pathogenic bacteria in oral cavity and pharynx. And the other function is, it helps during cell movement. But how? It helps as, for example, in embryogenesis, through the pathways of fibronectin, it guides cells to their destination. So now see the structure of fibronectin. It exists as a protein dimer consisting of two nearly identical monomers. You can see in the picture, there are two dimers you can see which are linked by a pair of disulfide bonds, and there are four fibronectin domains which attached, and these four domains allow fibronectin to associate with other fibronectin molecule. Now the laminin. It is most abundant glycoprotein in basement membrane. Domain binds collagen type 4, heparin, heparin sulfate. It helps in cell surface receptor, in cell adhesion, cell differentiation, anchoring the glycoprotein to basal lamina. Diseases associated with laminin protein is congenital muscular dystrophy, and it can be also in progeria. Laminin defect can also be there. Progeria means early aging. The next is the proteoglycans. You can see in this figure, proteoglycans consists of a core protein. This is core protein with chondroitin sulfate is attached here, keratin sulfate is attached here, and hyaluronic acid is the backbone to which these all core proteins are attached. So basically, proteoglycan composed of a core protein to which glycosaminoglycans are attached, and the core proteins are attached to long hyaluronic acid polymer. They have been classified and they vary in tissue of origin, function, core protein types. Examples of proteoglycans includes aggregans, syndecan, beta-glycan and seriglycan. How the synthesis of proteoglycans occur? So, it starts with core protein synthesis from ribosomes on rough endoplasmic reticulum. Here you can see the lumen of the rough endoplasmic reticulum from ribosome synthesis on the RER, and the addition of glycosaminoglycans takes place in the Golgi apparatus. And now the resultant protein was formed in this figure, you can see it is shuttled to the Golgi apparatus where it is glycosylated with the glycosaminoglycans within the Golgi apparatus itself. Now this completed proteoglycan is released into extracellular matrix via exocytosis. So first the core protein synthesis is taking place, then glycosylation is taking place, then proteoglycan is released. Elongation takes place as the units in the saccharide chains are elongated in alternating acidic or amino sugars. Then further modification takes place with the epimerization of glucuronic acid to iduronic acid, which is catalyzed by epimerases, and sulfation of the amine sugars are catalyzed by sulfotransferases. So, this is all about the extracellular matrix.